with this organism here. This is the budding yeast known as Saccharomyces cerevisiae. And it's actually been an incredible friend to mankind. Man has been taking advantage of this organism for several thousand years because it likes to take sugars and turn it into alcohol and CO2. And that means that we have used this organism to make bread, and we've used this organism to make beer and wine for thousands of years. Now, about 100 years ago or so, brewers were facing the problem that sometimes they'd brew a batch of beer and it would be great, and another time they'd brew a batch of beer and it would be terrible. And so they decided to start working on the biology of what was going on in these brewing vats of beer. And they found out that there was this particular organism that was responsible for changing the grain into alcohol and CO2. And they then started, more and more work was done on this organism, originally with this goal of brewing better beer. But it turns out that as biologists studied this organism more and more, we found out that it has a tremendous number of properties that make it just ideally suited for biological research. For example, it can grow either in the haploid or the diploid state. And we are obligate diploids. We have one set of chromosomes from our mother and one set of chromosomes from our father. Until that sperm and egg comes together, the sperm itself is the haploid, the egg is the haploid. They're not doing anything much. It's only when they come together that you form a new organism. But in yeast, they can live, and you wouldn't really know much difference between them being the diploid state and the haploid state. But for a geneticist, that's a dream. Because if you have one bad copy of a gene and one good copy of a gene, you can keep that bad copy in a diploid state so that the good copy takes care of the cell. Then when you sporulate the cells and you get the haploid progeny out, you can see what that mutation does. It's really a wonderful property. Yeast cells also have very small genome, so it turns out that this was the first higher organism to have its genome sequenced. Now you may be wondering why I would call this a higher organism. It doesn't seem like a higher organism, but it actually is. It has much more in common with human cells than bacterial cells do. And the reason is that these yeast cells have a membrane-bounded nucleus. They contain mitochondria, which are energy factories for the cell. They contain a whole series of membrane-bounded compartments where they put different proteins are compartmentalized and secreted from the cell or not secreted from the cell, moved around the cell in different ways. And that's a lot like our cells work. So even though these cells are separated from us by about 1 billion years of evolution, they actually do things in a remarkable number of details. They do them very much like we do. So we often use these cells as living test tubes to study really complicated problems in protein folding biology. And that takes us into problems of evolution. It takes us to the problem of human diseases.